Assalamu alaikum. Today's lecture is about instrumentation of Raman spectrophotometer. The different components of the spectrophotometer have been drawn on the board. We will discuss one by one each of them. The first component is the radiation source. Initially, before till early 1960s, classical radiation sources were used. For example, mercury arc lamps were used, which provided a wavelength radiations of 435.8 nanometer wavelength, and xenon lamps were used. But later on, in the late 1960s, these radiation sources were replaced by lasers. Lasers are more efficient than the mercury arc lamp or xenon lamps due to the following facts: that they have a small form factor, they have low power consumption, they give us stable power output, and they give us stable wavelength output. Now, different type of lasers have been commonly used. Examples are: number one, argon ion laser, which provides us radiations of wavelength 488 and 514.5 nanometer. Second laser source is krypton ion lasers, which provide us 530.9 and 647.9 nanometer wavelength radiations. The third is helium neon laser, which provide us with a radiation of 632.8 nanometer, and near infrared laser, which provide us with a wavelength of 785 and 830 nanometer. Now, the wavelength, the near IR lasers with a wavelength of 785 nanometer and 830 nanometer are commonly used. Now, lasers are important to use as radiation sources because they have two advantages. Number one, laser has a narrow band which lead to sharp peaks. If the bandwidth is not narrow, if we have a broad band, that we will get wide peaks. Secondly, it is very rich in photons. We know that the Raman scattered radiations give us very weak signals. Therefore, we must have very large number of protons to observe the Raman scattering. These lasers are then passed through a polarizer, and the function of this polarizer is that they give us linearly polarized light or plane polarized light, which is vibrating in a single plane. The second component of a Raman spectrophotometer is the sample. The main advantage of Raman spectrophotometer is that we can take the sample, the liquid sample, in any type of vessel. For example, we can use cuvettes, we can use glass cells, we can use plastic bags, or even we can take the sample in transparent wrappers. Now, the laser radiations, after being polarized by the polarizer, they pass through the sample and are scattered. Now, we already know that majority of these radiations are scattered with no change in frequency, and we get Rayleigh scattered lines, or we get Rayleigh lines, but some of them are also scattered with a change in frequency, and these are known as Raman scattered lines, which may be Stokes lines or anti-Stokes lines. Now, these scattered radiations, they are passed through two lenses, convex lenses, in order to converge them onto the filter. Between the two lenses is another polarizer, which is called analyzer. There is another component which is known as analyzer. The scattered radiations which have been converged by the two lenses, they are focused on a filter. The function of this filter is that it will eliminate the Rayleigh lines and only allow the Raman scattered radiations to pass through the filter and strike the diffraction grating. Now, the scattered radiations, after being filtered by the filter, they enter the next component, which is the diffraction grating. The diffraction grating disperses the radiation at different angles depending upon their frequencies. Now, on the basis of this component, the Raman spectrophotometers may be of two types. They may be either dispersive spectrophotometers which use a diffraction grating, or they may be non-dispersive Raman spectrophotometers which use Michelson interferometer rather than diffraction grating. Now, if we are dealing with dispersive spectrophotometer which use diffraction grating, then these radiations which have been dispersed by the diffraction grating pass through a slit and they enter the last component of the spectrophotometer, which is called detector. Now, the detectors may be of two types. They may be photomultiplier tubes or they may be charged coupled devices. Photomultiplier tube is a single point detector, that is, it can detect the radiations at a single point. Therefore, if we are using photomultiplier tubes, then the diffraction grating must be rotated so that the radiations which have been dispersed by the diffraction grating enter the slit and the photomultiplier tube at that specific angle, so that the diffraction grating must be rotated to get these rays directly into the photomultiplier tube. On the other hand, if we are using charge coupled devices, then these charge coupled devices, they are known as array detectors, and in this case there is no need to rotate the diffraction grating and we will get the signals directly into the charge coupled detectors. Now, every instrument, Raman spectrophotometer, has a resolution. The resolution of the spectrophotometer means how much it can separate the different peaks, different Raman scattered peaks. Now, if the instrument has high resolution power, then the peaks will be separated from each other with a wider distance, and we can only study the photomultiplier tube can detect only a part of the Raman spectrum and cannot cover the whole spectrum, which will be very wide spectrum. However, if the spectrophotometer has a low resolution power, it means that the peaks will not be very much separated from each other, they will be closer to each other, and the whole spectrum can be detected by the photomultiplier tube or by the charge coupled detector. So, if we want to study a particular part of the Raman spectrum, we will use high resolution, and if we want to study the whole Raman spectrum, we will use low resolution instruments. Allah Hafiz.